All right, blood. I don't have a song for this. Oh, my dad joke. Why don't ants ever get sick? Because they have antibodies. Har har. That'd have been a better one to do during lymphatics, but it's the next one that came up.
Are they all science jokes or no?
No, I pick and choose. Because I'm still in March as far as tearing off the days of the calendar, so I have a lot to get up to where I.
We're gonna have to do 2 then.
No. Some of them are like dad jokes are bad, but some of these are just like why are these even written? All right, blood, not a system in and of itself. It's technically part of the cardiovascular system. All right, and its primary function in that system is for transportation. Right, heart pumps the blood, blood reaches the tissues through the arteries, comes back to the heart through the veins. Know that between what I knew I was missing something. When you're down at the capillary level, you have exchange of substances out of the capillary with the interstitial fluid. So since blood is always thrown through capillaries, you have this exchange. It's responsible for maintaining interstitial fluid stability. And that's one of the ways that we maintain a constant 0.9% saline solution throughout our body. Also distribution of heat. Okay, so the core of our body, right, is warmer. We pump blood out of the warmer area to colder areas. It gets cooled down, then comes back to get heated up. All right, what's in blood? It's about 8% of our body weight, so if you weigh 100 pounds, it's about 8 of your pounds. The average adult, about 5 liters of blood, so 2.5, 2 liters of Mountain Dew. I don't know why I always use Mountain Dew as the go-to, but because everybody loves it, I don't drink it anymore. All right, blood is 45% cells, so slightly less than half of your blood, even though it's the cells that make up the red part of your blood, is only actually cellular. Most of that is white blood cells, but then you have white blood cells and platelets, all right, and we're going to talk about the functions of those as we go on. The rest of it's plasma, all right, and plasma.
Did you even say most of that's red blood cells? You said white blood.
Did I say that?
Yeah.
Yeah, most of it's red blood cells. Thank you for catching that. If I do say stuff like that, because I just go on autopilot mode sometimes. Plasma is water plus everything dissolved in it. It doesn't matter what's dissolved in it as long as it's dissolved in it. And we'll talk about some of these things as we go on. Now, I don't know if I have this in here, but there's also something called serum. You guys heard of that, right?
Yeah.
Anybody know what serum is?
Plasma without platelets.
Right, plasma without the clotting factors. All right, so if you have a tub of plasma, you let it clot and take out the fluid, that's serum. So in science fiction movies, everybody talks about this new serum that's going to change the world. That's their different definition. All right, good summary slide to go back to. What are all the things in blood? All right, so it's that all the cellular components are generally called formed elements of the blood because not all cellular components are full cells. Okay? Plasma contains everything else. We talked about a little bit about that, and we'll go through all of these as we go through this lecture packet. All right, so we'll start with plasma. Most of it's water. You have plasma proteins, all right, and those are involved. Actually, we talk about all these in detail in the next couple slides. All right, so we'll talk more about that. Gases and nutrients, we talked about O2 and CO2, our main gases that are transported and released or taken in at the respiratory level. And waste. breakdown products of different, bless you, different metabolites in our body. All right, and then of course our electrolytes. We're not getting into hypernatremia, hyponatremia, hypercalcemia, hypocalcemia, but the electrolytes are in there as well. Assume that's a cell. What electrolytes are high outside of cells?
Sodium and chloride.
Sodium, chloride, one more, big one, important one.
No, potassium's inside the cell.
Magnesium.
Oh, calcium.
Calcium. Also, make sure you put charges on things because none of them exist that way. So that's going to be important to know, I think, next week's lecture. But our blood will carry that around, and it's because our blood carries those around and stuff comes from our diet that allows that concentration of those to maintain equilibrium with the interstitial fluid. All right, so what are the plasma proteins? Most of them are albumins. All right, albumins, their primary job is to maintain osmotic pressure. So osmotic pressure, when we get into the capillary level and we have a bunch of proteins, mostly albumins, in our capillaries, that draws fluid back in. All right, so that's the whole basis of why when we push everything out of capillaries, our capillaries don't run dry, because the osmotic pressure pulls fluid back in. Also binds and transports molecules. We talked about that when we talked Wednesday, about steroid molecules. They will bind to albumins or other plasma proteins to carry lipid-soluble substances. Fibrinogen, and notice it says other clotting proteins as well. Most of your clotting factors are made in the liver, and then they get secreted into the blood, and they just kind of circulate around waiting for an event to happen. What would that event be? Yes. Some kind of tissue trauma. Yes, paper cuts would be a good example. All right, globulins. We have 3 flavors of those: alpha, beta, gamma. Gamma globulins, that's where we're going to start. Those are going to be our antibodies. Antibodies. And we'll talk more about that when we get to the lymphatics, but they're, of course, important in fighting infection. Before we even get there, what do antibodies do? They bind to antigens. Do they destroy things?
No.
Do they do anything really important to get rid of things? No, they simply bind, and they serve as like a flag. All right, alpha and beta globulins, they are also produced by liver, and they transport lipids and fat-soluble vitamins as well as albumin. So alpha, beta globulins plus albumin, those are transport vehicles. Gamma globulins are antibodies. Albumin is our osmotic protein. All right, the only thing you're going to see this again, but the importance of this is we have plasma right here, all right, that's a fluid portion of our blood. Across the capillary membrane, it is in constant contact with interstitial fluid, so anything that happens to the plasma is going to have direct impact on the interstitial fluid. And then the interstitial fluid is then connected to intracellular fluid. So whatever happens in interstitial fluid is going to influence what happens in intracellular fluid. All right. Likewise, let's say we take in a bunch of water, for example, and that goes into plasma. It's going to get into the interstitial fluid, and it's going to fill up our cells as well, right? If we lose fluid, if we lose water from the plasma, that needs to be replaced. Where's it going to pull from? Interstitial. If interstitial loses water and becomes more concentrated, it's going to want to regulate itself, so it's going to pull from the cells, and then the cells will get dehydrated. So there's this constant back and forth between the plasma and the interstitial fluid. I'm sorry, intracellular fluid through the interstitial space. And that's going to be an important concept to remember. Not necessarily for blood, but definitely for fluid, electrolyte, and acid-base balance. All right, so let's start with red blood cells. I don't have a song for today. I do, but nobody would like it.
Raining Blood by Slayer.
Do what?
Raining Blood by Slayer.
Never heard of that one. It's called Blood by In This Moment. Anybody ever heard of that one?
Yes.
Yeah?
Meadowlands.
Pardon?
They're playing at Ottawa Tavern. I think in August or July.
Really?
I think so.
I don't mind concerts as long as I can sit down and have a beer. I hate going to concerts and just standing there. I watch my watch the entire time. All right, red blood cells, also known as RBCs, erythrocytes. They are small biconcave discs. So what that means is that they are caved in on both sides, but then there's this ring, so they look like a donut without the actual hole. The main importance of them is they contain hemoglobin. And what does hemoglobin do? Bind oxygen. Good. That makes up 1/3 of the total cell volume. So 1/3 of your erythrocytes is hemoglobin. And in a couple slides, I'll equate that to an actual number of hemoglobin molecules. Note that mature cells don't have DNA. So what does that mean they don't do? Divide or make new proteins. So once a red blood cell is formed, it's got everything it needs for its lifespan. And what is the typical lifespan of a red blood cell?
90 to 120 days.
Days. Okay. They also lack mitochondria. All right. So they can't produce much energy, only through glycolysis. So that doesn't, if you remember, produce much in the way of ATP. All right. Why is it biconcave? Why is this donut shape so important? What it does actually is it brings the hemoglobin close to the cell surface. All right, and if you bring the hemoglobin close to the cell surface, what does that decrease? Decreases the distance for diffusion. All right, so that means oxygen can be transported back and forth much easier because they're placed so close to the cell membrane. All right. The other thing it does is it allows the red blood cell to squeeze through small capillaries. So a lot of capillaries in your body are actually smaller than red blood cells. So what this donut allows you to do is kind of fold over, squeeze through, and then boop, pop back to normal shape. All right, lifespan's only about 120 days. 30 times 4 is 120, so about 4 months. All right, clinically, these will be your values. Am I going to hold you responsible for them? No. At some point, will you be held responsible for them? Of course. Here's your first introduction for them. What I do want you to know, though, is that when you go from children to women to men, the number of red blood cells increases. So men have more red blood cells simply because they have more muscle mass on average. And that muscle mass needs to be supplied with oxygen. All right, hemoglobin. All right, that's a normal concentration. We'll never ask you that. Oh, it says 34 grams per 100 milliliters of packed cells. Another definition of packed cells is our hematocrit, and we'll talk about that in the next slide. And hematocrit normally is about 40 to 45% for men and women. So if we say that blood is made up of formed elements in plasma, and those formed elements make up about 40%, then hematocrit should also be about 40%. Remember that a couple previous slides ago. Okay, just some statistics about it. The other thing I wanted to say about hemoglobin is its structure. There are four polypeptide chains. Polypeptide chains are made up of chains of what? Amino acids, right? Amino acids. Each one of those polypeptide chains has this heme group, and each heme group has a iron. 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 I-R-O-N. Iron. Has a ferrous atom. How about that? Nerd. Talking about me, not you. All right, going to hematocrit really quick. This is your hematocrit. Clinically useful tool. What they do is they take a sample of blood, they put it in a centrifuge, spin it down. Here's your red blood cells. Here's your plasma. And sometimes you can see right around in here a thin white layer or milky layer. That's called the buffy coat. Those are our white blood cells. Okay? With a hematocrit, that packed cell volume, 99% of that's the red blood cells. That about 1% is the white blood cells. Everything else is fluid. All right, so if you take total volume and you take the volume of red blood cells, red blood cell over total volume, that'll give you your hematocrit. All right, so what percentage of your total volume of blood is actually red blood cells? All right, so normally 40 to 45%. However, certain conditions can increase or decrease that hematocrit. So if this is normal, let's assume that is 40%, this would be anemia. What happened to your hematocrit? Did it increase or decrease? It decreased. All right, so maybe this is 20%. And what do we know about anemia? You're lacking what? Red blood cells. Doesn't have to be iron. There are many different types of anemia. We talked about one that's related to what? Intrinsic factor in B12, right? All right. Polycythemia. Anybody know what that is? Overproduction of red blood cells, right? Okay, that's a higher hematocrit. Let's say that's 75%. All right, notice that it takes up more of the volume of the total blood than normal. All right, what's the problem with anemia? Why is a low hematocrit bad? Right, you can't transport enough oxygen to tissues. What's the problem with a high hematocrit?
There's too much to transport.
Maybe if they're,
Okay, too much to transport.
if red blood cells are all competing for oxygen, there's going to be less overall oxygen exchange. So,
Good guess, but no.
Too thick?
Too thick. All right, you're going to have too many red blood cells. They're all going to be competing for space. It's going to be too thick. So that means it's going to be harder to push. That's going to drive your pressure up. All right, that's why it's bad. So if we had somebody who's dehydrated, which one of these hematocrits would be most represented by that?
What did you say?
Dehydrated. 3. It'll be 3. Because, not because you have more red blood cells, but because you have less fluid. Okay? Does that make sense to everybody? See, senor. All right, hematopoietic stem cells. These are stem cells in our bone marrow. Okay, red bone marrow specifically. Not yellow bone marrow. What's yellow bone marrow made of? Adipose tissue. Okay? And these are stem cells from which all cells of the all formed elements of the blood are derived. So every cell that's in your blood, every formed element is going to come from red bone marrow and these particular cells. All right? What happens is, even though these are stem cells, they have the potential to become many different things. It's hormones that bind that tell them what kind of cell to differentiate into. Okay? So they respond to hormones and growth factors. Outside of red bone marrow, these are other places that they can be found during different stages of development. All right, now it turns out that over your lifespan, where red blood cell production occurs changes. All right, initially, early in life, that's what these are showing, it's going to occur in the shaft of your tibia and femur. Why those places? They are the biggest bones in your body. Okay? Then, as these get bigger and bigger and more mature, they get replaced by yellow bone marrow. And they're not going to produce red blood cells anymore. So, that job has to be shifted over to trabecular bone. All right, so bones that have mostly trabecular structure, also known as spongy bone, right? So that would be in our vertebrae, sternum, ribs, also ilia. Okay, and that's where they remain kind of through the rest of our life. And notice that that slowly declines. So as we get older, red blood cell production actually declines. All right, I like this picture because it kind of shows the overview of how hematopoietic stem cells work. Here would be your stem cell. Here would be all of the differentiation pathways. This would be RBCs. This would be platelets. These would be our granulocytes, and then our agranulocytes. Okay, so what I have here is if a hormone called erythropoietin, which we'll go into a little bit, binds to this cell, then it will start dividing and produce cells down the red blood cell differentiation pathway. If thrombopoietin stimulates the cell, it will go down the platelet pathway, and so on. I didn't mention ones for these because there are a lot more of them and they're a little more complex. Okay, so be aware that it's a hormone binding to receptors on this cell that will cause it to differentiate down a very specific pathway. All right, so what is that hormone doing? So I'm getting to this question right here. What is that hormone doing? Question.
On specific genes
Good, okay,
to go down a certain pathway, like the SRY gene.
right. So my question was supposed to be, what is that hormone doing to the cell, turning on genes, right? All right, but our discussion right here is about red blood cells. So this is the pathway of a red blood cell, and I'm not going to necessarily hold you responsible for the different stages of development, but I put this here because it's going to come up in a couple different areas as we're going on. Notice that when a red blood cell starts forming, it does have a nucleus, all right? And as it matures, it loses that nucleus. It just spits it out because it doesn't need it, because that does nothing for its actual function, all right? Also with the mitochondria, it will have mitochondria. Those either get spit out or degraded, and then you end up with a biconcave red blood cell that carries oxygen, all right? If this pathway doesn't work properly, for one reason or another, you can end up with anemias, and these are different types of anemias. One is iron deficiency. Cells will end up microcytic. What does that mean? Small. And hypochromic. Low.
Variability, like cells.
Nope.
Iron.
Nope. Color. Low in color. So they'll be small and colorless. All right, and that's a characteristic you're going to see with iron deficiency anemia. With sickle cell anemia, that is a genetic problem, right? And that genetic, and we'll talk a little bit about it later, produces a sickle shape in the red blood cell. And not only can they not carry oxygen properly, but that sickle shape causes them to get stuck in capillaries. And if they get stuck in capillaries, blood coming behind it will get stuck as well. You don't have perfusion to the tissue, and then you have a sickle cell crisis. All right, if you have a B12 or folate deficiency, that will result in a megaloblastic anemia. You're like, wait a minute, but you said it was a pernicious anemia. Pernicious anemia is a type of megaloblastic anemia. All right, and notice the morphology of that. They're all over the place. You have hypochromatic. You don't have microcytic, but you have red blood cells in various stages of development. So if you compare that to what you see over here, it looks like they're developmentally messed up. And then erythroblastosis fetalis. If you So this can happen as a result of a mismatched blood transfusion or Rh factor, like your baby is Rh positive, mom is Rh negative. For the second baby, mom's antibodies will attack the baby's red blood cells because they had another Rh positive baby before, and that will do this to the baby's blood. All right. I think we talk more about that later on. All right. So, about the function of red blood cells, there's a negative feedback loop. All right. If we have a decrease in blood and oxygen, the kidneys and to some degree the liver will detect that. All right. There are receptors in the kidneys and livers that will detect changes in oxygen. The kidneys will release erythropoietin, and we talked about that early on. That was part of the hematopoietic stem cell. That will go and stimulate these hematopoietic stem cells to form proerythroblasts. They will eventually differentiate into red blood cells. They'll go into the circulation and increase tissue oxygenation. All right, so low blood oxygen, you do something to create more red blood cells that will increase blood oxygen. All right. As O2 increases, that will feed back to the kidney and shut off production of erythropoietin because you don't need it anymore. What would happen, theoretically, if you keep stimulating erythropoietin? If erythropoietin keeps stimulating hematopoietic stem cells, what would happen?
It'll make too many blood cells.
Pardon?
It'll make too many blood cells.
Okay. And what's that called?
You'll become an
Polycythemia. What'd you say?
I said you'll become an Olympic runner.
He'll become an Olympic runner. Actually, so what was it? It was cross-country skiers were known for blood doping. They would take out their blood, freeze it. When it comes to race time, they would inject all those bloods back into themselves to give more oxygen, give them an advantage. The problem is, as we discussed, it makes blood thicker. Some of them died because that blood was too thick. They were working themselves too hard, and it's totally worth it. Because we eliminated those cheaters from society. I'm kidding. All right. Some factors that will decrease oxygenation: low blood volume, anemia, low hemoglobin, poor blood flow, pulmonary disease, all those things you normally think of as decreasing blood oxygen. Also exercise as well, right? Exercise will cause a decrease in temporary decrease in oxygen, which your body will make up for, but not through this process.
I have a question.
Yeah.
Do like COPD patients have like thick blood then?
No. They will, generally their oxygen saturation will just be lower, and they will operate at that level. So their body will just adjust everything to operate at that level. Theoretically, for a temporary period of time, they might, but then that will equilibrate over time. All right, so going into a little more detail about this, if we are deprived of oxygen for minutes to hours, and I'm not talking like zero oxygen, like you die, but I'm talking about low oxygen levels, that's going to lead to an increase in erythropoietin. However, we don't get new circulating red blood cells until about 5 days later. So this is a long-term thing, not a short-term fix. What would be a short-term fix for reduced oxygen levels? Right. Breathe more. Shut up. That was never mind. All right. So I wrote here implications for blood donation. If this takes 5 days and you are low on oxygen, they're going to be like, all right, we need to get more red blood cells in right now. Let's give a unit of blood. Okay. So when we throw erythropoietin into the mix, that increases the production of red blood cells and can accelerate the process up to 10 times, okay, above normal, above basal rates. Okay, and then of course that will remain high until oxygen is restored, but again, there's going to be a 5-day lag. So if you start seeing an increase in oxygen after 5 days, that reaches normal levels after 7 days, then it's after 7 days that'll actually shut down. Okay. All right, what kind of dietary factors are important for making red blood cells? Well, we've already talked about this one, vitamin B12. Now we're talking about why it's important. It is absolutely required for DNA synthesis. All right, and you're like, well, don't red blood cells not have DNA? Remember, in that pathway, they do have a nucleus and DNA up until the red blood cell extrudes it. And that's important for all the things that need to go into making a red blood cell what a red blood cell is. And then it's like, all right, we don't need you anymore. Get rid of it. We know that B12 is carried by intrinsic factor released by parietal cells. Where is B12 absorbed? The ileum. Good. Excellent. All right. Folic acid, another important vitamin, also required for DNA synthesis. Okay, so very similar to vitamin B12. And we talked about a couple slides ago, if you have lack of vitamin B12 or lack of folate, you get megaloblastic anemia. Iron also required, of course, because that's important for hemoglobin synthesis. And vitamin C is important not for the production of red blood cells, but for making sure that we get iron absorbed from our diet. Okay, without vitamin C, we don't absorb as much iron. We could develop an anemia from that as well. That would be iron deficiency anemia. All right, how is hemoglobin formed? So it occurs from this stage to the reticulocyte stage. So in other words, every stage until the nucleus is thrown out. Okay, once the nucleus is thrown out, notice that reticulocytes will still have a small amount of ER. And what's ER important for? What does it make? Proteins. And that will help support hemoglobin synthesis. Why? Because you need to make them those globin chains as well. Okay, so it's important for that purpose. All right, all I want to say about this, as far as hemoglobin goes, it is a complex chemical process. I'm not holding you accountable for that, but this is what hemoglobin looks like. All right, and it's this molecule that gets incorporated into each of those 4 polypeptide chains. All right, which will have an iron sitting at the center. All right, and what's the charge generally on iron? Let's just say positive for now. Okay, it depends on its oxidation state, but it's positive. When it binds oxygen, what kind of oxygen is it binding? Is it binding that? Or is it binding O2? O2. All right. Does O2 have a charge? No, but it has a dipole, so it has a slight negative charge. Therefore, that slight negative charge will interact with this strong positive charge in iron. You guys didn't think you were going to hear chemistry terms, did you? Oh, did you guys hear? I was only in it for one. Gavin was talking about physics. Like, physics is important. don't want to know. All right, so specifically, most of the globin chains that we have that make up hemoglobin are 2A and 2B, which make up what we call hemoglobin A. Okay, that's the predominant form that we have. Before birth, we have a slightly different hemoglobin that has a higher affinity for oxygen. And over time, as we age, that changes slightly. We're not going to go into the details of that, but just know that's different before we're born and that it changes slightly as we get older. Okay, and we've already talked about all of this as well, so I'm not going to go into any more detail about that. Oh, what? I edited this last night. 30 microliters of hemoglobin in 90 microliters of red blood cells. Remember, hemoglobin takes up 1/3 of the red blood cell volume, so those numbers add up. And, oh God. In one red blood cell, I'm forgetting a number. I think it's like 150 million hemoglobin molecules. 250 million. Is it in your notes? Or you just remember? Okay. So 250 million. So when we say that a red blood cell is dropping off, has 4 oxygens, it's like an average thing we're talking about. It's not that it only has 1 hemoglobin molecule with 4 oxygens. There's 250 million that are doing this job. So just to give you kind of a scale comparison.
So it's 2 billion oxygen atoms per red blood cell.
Yeah. I'm glad somebody's doing the math. Yeah, actually, so think about that. When we talk about capacity to bring oxygen, we have a way overabundance. All right, so we have a huge reserve. So when something goes wrong, it has to really go wrong in order to reduce oxygen carrying capacity. So one specific example where we see differences in the globin chains is in sickle cell anemia. So this is where we'll talk about that more specifically. What sickle cell anemia does is in one of the hemoglobin chains, it changes a glutamic acid to a valine at amino acid 6. And that's based on one single point mutation, so change in one base in the entire gene that leads to this. So you take something that is a negatively charged amino acid and you change it to something that's not charged. That's going to change the way that hemoglobin, that globin chain folds. Okay? So if you change the way that it folds, that's going to change the way the red blood cell is shaped, since there are so many hemoglobin molecules in that red blood cell. Okay? Especially when exposed to low oxygen. Mostly this will happen when you're exposed to low oxygen environments. So when that does happen, again, this is kind of what we mentioned earlier. Those red blood cells will get caught in smaller capillaries, cause things to get bunched up. You have lack of perfusion to the end organ or whatever that capillary is supplying, and it results in lysis of the red blood cells, so they'll all start to break open, and of course vascular occlusion, which is what that is showing right there. So both are bad, right? Because you're releasing cellular contents into circulation that shouldn't be there, and that will trigger an immune response, and it will block blood vessels. All right, we've already talked about this. I'm going to go through this really quickly. Oxygen binding to hemoglobin, it must be loosely bound because it has to come off, right? It has to be loosely bound because it has to come off. So there has to be a sweet spot of affinity for oxygen. All right, when oxygen concentration is high, which is where's oxygen concentration high in your body? Alveoli, in the lungs, right? hemoglobin holds on to oxygen. Where we have lower oxygen concentration, big toe, because that's as far as you can possibly get, then it's going to release O2. But it's not just because it has a low oxygen, it's also because you have an increase in what else? CO2, an increase in hydrogen, which equates to a decrease in pH. You may or may not have a higher temperature. Okay? I'll put it to you that your big toe is going to be lower in temperature than your lungs. Question? All right, so let's talk a little bit about the iron that's in there. All right, we know iron is important for putting into hemoglobin because it's the thing that binds molecular oxygen. And because of this, we store iron. It's very, very tightly regulated. So we have
Just take a moment.
I get distracted easy. The storage of iron is highly regulated. We have multiple storage forms, and each of those operates slightly different from each other. Most of our iron is in hemoglobin, so it's taken up, it's doing something. All right, that means 35% of the rest has to be doing something else. 4% of that 35% is in myoglobin. What is that? Right, it's the specialized form of hemoglobin that's in muscle. 1% is in intracellular heme compounds, so being put into things that are going to become red blood cells. Less than 1% is associated with circulating transferrin. So transferrin is a storage form as red blood cells are circulating. And about 15 to 30% is stored as ferritin in the reticuloendothelial system. What that is phagocytes in our red liver, spleen, that remove red blood cells from circulation and recycle all the parts of the red blood cell. Okay? So these macrophages store it as ferritin inside themselves and will release it to circulation so that it can be recycled to new red blood cells. All right. Looks complicated. It's really not. We get iron from our diet. That goes into our plasma with the help of vitamin C. Right? And that's how it's transported to where it needs to go. Notice that it's transported to red blood cells, and notice that it's transported to tissues. All right, so it will go from plasma to either of those places. If it's going here, where is this actually going? So if transferrin is taking iron to make red blood cells, where is this actually occurring? Where do you make red blood cells? Bone marrow. Okay? After about 120 days of circulating, the reticuloendothelial system will get rid of the red blood cell. Those macrophages will free up the iron. We'll talk about this a little bit later. Free up the iron. That free iron can then be stored in tissues as ferritin, which is highly reversible, so that can go back and forth readily. It can be stored in some tissues as hemosiderin, which favors the formation of hemosiderin, but it's insoluble, so it's very difficult to get it back into free iron. All right, so two different storage forms, but they operate on much different timelines. And then that free iron can go into heme, like we said, the 0.1%. And then some enzymes rely on iron to work. So as those enzymes are degraded after a certain time of use, that will free up some iron as well. So that is what this thing is showing you. And also notice that blood loss can be 0.7 milligrams of iron a day in menses on average. which was that fun lecture on Wednesday. All right. So, where do we get iron? It's absorbed in the small intestine. It binds to something called apotransferin. When iron binds to apotransferin, it turns into transferrin. So, apotransferin doesn't have iron bound. Transferrin does have iron bound. Otherwise, it's the same molecule. Okay, and that's this molecule down here that we were talking about on the previous slide. All right, iron can theoretically be released to any cell, but it's going to be released to cells that are going to be in most need, and mostly that's going to be red blood cells, so that you can make new so bone marrow. All right, red blood cell precursors. So go back. All right, too many slides. These cells have receptors for, where is it? Have receptors for transferrin. So that's how transferrin knows where to dock and release the iron, because the transferrin binds to these receptors, and then the iron gets endocytosed and then processed. All right, so what about hemosiderin? Most of our iron is stored as ferritin, all right, intracellularly, and releases fairly easily. I talked about that. Hemosiderin, that is a really large cluster of iron that's held in check by a series of proteins, and it's not soluble. All right, it's not soluble, so it's harder to get back out. And if it accumulates to too high of levels, it can lead to problematic health conditions, of which we're not going to discuss. Okay, so hemosiderin can be bad. Ferritin, not so much. Where are we? Let's get through iron. Yeah, let's get through iron, and then we'll take a break. All right, when iron in the plasma is low, all right, so this is kind of like that oxygen, iron is released from that storage form inside of cells, bound to transferrin, and transports into blood. Okay, we saw that in previous slides. Of course, then that transferrin will take the iron to bone marrow, right, through receptors. The cells will internalize it and deliver it to the heme molecule. All right, so that's kind of how that process works. Iron is low, ferritin puts it to transferrin, bone marrow, receptors, internalized, and to heme. If we have loss of the plasma transport protein, that can lead to hyperchromic anemia. Why? Because you're not getting iron into the red blood cells. Iron is what gives them the color. Because really, what's our blood? If we consider that iron is a primary component of red blood cells and gives it its color, really our red blood cells are just buckets of rust. Okay? When red blood cells are destroyed, so senescent, when it reaches that end of that 120 days and it's just exhausted and it needs to be removed from circulation, we're not going to go into the physiology of that. That is probably a week's worth of lectures in and of itself. They're ingested by macrophages in, remember, the RES system, located in the spleen and liver. Okay? So then that's going to release the iron for storage. All right, what's iron balance dietary-wise? In men, we lose most of our iron in the gut, right? We poop it out. Females lose more because they menstruate so GI plus menses. So you actually, if you add all that up, you actually lose more in your menses than you do from the GI system. Okay? From pooping it out. Okay? Yeah, we won't worry about that. Except for this last part, sorry. For absorption, the maximum we actually absorb is a couple milligrams per day. But that can increase 5 to 6-fold if our body needs it. So if we run out of storage, our body will actually absorb more until we reach those storage capacities, and then it will shut down and we won't absorb as much again. Okay? So let's take a break there and move on after 5 minutes.
No, yours is purple. Wait, why is yours a different color? That's so fun.
Oh, so yeah, if you notice different colors on your devices, like students pointed that out to me last year that sometimes the devices change the color of some of my pictures. It's a weird thing. I don't know why. So if you have a purple liver,
No, I don't like
okay. So let's talk about red blood cell destruction. 120 days. Things are worn out. Your red blood cell is low in energy. Cell membrane is becoming degraded because it's squeezing through small capillaries. The macrophages in the reticuloendothelial system in the spleen and liver will detect that, and it will destroy those cells. Was that a hiccup?
Yes. Yeah.
Okay, so here's what that looks like. Here's our red blood cell. It gets broken down into the globin. That gets recycled as amino acids. Heme, that will be converted to bilirubin, transferred into blood by albumin, go to the liver, conjugated with glucuronic acid, which will make it water soluble, and then that will be secreted into bile. So that's how we get from a broken down red blood cell to the color of our poop and urine. Okay? And then the iron, then, of course, we talked about where that is going to go and get stored as ferritin. Okay? So that senescent red blood cell gets broken down into those components by the macrophage and will go to places where it needs to go. Okay. White blood cells, leukocytes. Bunch of different types. They do different things. This is going to be a survey of them, so not going into a whole lot of detail. We have two families, the ones with granules or, the ones with granules and the ones without granules. Okay? What are the granules? Anybody know? No. What are the actual granules in those cells? Nope. Things to be secreted. So these are full of secretory vesicles, and their job is to secrete stuff mostly. So the first type and the most highly abundant are your neutrophils. Okay? Generally speaking, they're the first to arrive at the site of infection and will take care of most of the infection before other white blood cells get there. And they can take care of most of foreign invaders, bacteria, fungi, and viruses. Eosinophils, they dye red with eosin. Eosin is just the name of the dye, so they like eosin. I don't care that you know the specific percentages, but what's the most abundant? Neutrophils. I care more about that. I care more about the functions, too. Eosinophils, they have a bilobed appearance. That's how you can pick them out. They're red and bilobed. They generally protect against parasitic worm infections. So if you see in your patient's chart you have an increase in eosinophils, first thing you want to think of is either allergy or parasitic worms. And then that will help you modulate your treatment. Basophils. They like basic dyes. They stain very dark. They also have two lobes. So how do you tell the difference between the two? One is red, one is dark. They circulate, but they also go into tissue and stay there for longer periods of time. And what they do is they release histamine. All right, histamine is one of our inflammatory molecules. So it releases histamine. That opens up blood vessels, gets more blood going to the area. They also release heparin. That inhibits blood clotting, so you can have a continuous flow of blood to that area, bringing in more white blood cells and more inflammatory factors. All right, so if you will, all of these granules in the basophils, histamine and heparin. Okay, they will release those and they'll degranulate. Agranulocytes, first are monocytes. They're a moderate-sized population in our blood. They are the largest cells in the blood, so out of all the white blood cells, they're generally going to be the biggest, so they're easy to pick out. And when they leave the bloodstream, they become macrophages. So essentially, monocytes are macrophages, but it depends on where they're located. They're not going to do much eating in the bloodstream. It's outside that they do it.
What's the arrow pointing to? What type of blood, red blood, white blood cell is that?
Don't know. I pulled this from a textbook, and they were trying to point out something else. Yeah. So that's irrelevant. Good question, though. All right, the other agranulocytes and the most abundant agranulocyte would be our lymphocytes. So those would be our T cells and our B cells, which we know are part of our immune system. All right, and when we get to lymphatics and immunity, we'll talk about those in more detail. Okay, so those are going to be the most abundant agranulocyte. Neutrophils, the most abundant granulocyte. When we talk about leukocytes, mostly you would say their function is protection against foreign invaders. Okay, so when we look at white blood cells and we see increased counts, what's our first thought? Infection, right. So we can do that a couple different ways. One, we can look at a smear. This would be normal white blood cells. You can see, and you can count them in a microscope. Anybody ever did that in your A&P labs in undergrad? Okay. Or you look at a high white blood cell count, you can literally just see and count more white blood cells. So that's the number of total white blood cells that should be clinically. You also do differentials. All right, when you talk about a differential, you're looking at the different numbers of different types of white blood cells. And I say see previous slides because we know that what their general population should be. So if you see like a lot more basophils, they make up less than 1%, and you see maybe 10%, you know you've got an issue. Okay. All right, and these, I'm not going to hold you accountable for those numbers. They just give you some reference ranges for what different types of white blood cells should be in terms of counts. All right, this will come up again in your clin med classes. Not relevant for this class. All right, the other type of white blood cell is a platelet, also known as a thrombocyte. Clotting cell is what that means. Okay, so if you go back to that picture earlier on where it showed the hematopoietic stem cells, thrombocytes come from giant cells called megakaryocytes. And these are really interesting because they will get created, they will sit on the outside of a capillary, kind of put their arms into the capillary, and as blood flow is going by, they break off. And so that's how platelets are formed. I thought that was really interesting. So we know that platelets, they're involved in the stopping of blood, and we'll talk more about that later on. I don't want to get into any more detail about that right now. All right, when white blood cells are created, of course, we know they are made in the bone marrow by hematopoietic stem cells, and they typically stay there until they're needed. Okay, they stay there until they're needed in the blood. Usually, there are 3 times more in your bone marrow than is in the blood, and that equates to about a 6-day supply. So at any given time, we have a 6-day supply of white blood cells sitting in our bone marrow. Okay, that's for granulocytes and monocytes. For lymphocytes, they develop in, they're made in the bone marrow, but then they go to the thymus, they go to the spleen, they go to lymph nodes and other lymphatic tissue to mature. Okay, and then they will stay there and wait for infections to come by. We'll talk about how lymph nodes work later on, not today, but when we get to the lymphatic system. They sit there and wait for foreign invaders to come by. And then megakaryocytes, they develop, they reside in the bone marrow, and it's the blood that's flowing through the bone marrow that they will sit on the outside of the capillaries, put some fingers into the capillaries, and then slough off as blood flows by. All right, lifespan of these white blood cells, granulocytes, if they're circulating in your blood, about 4 to 8 hours. So if they only last 4 to 8 hours, what does that tell you about the creation of white blood cells? All the time. Okay. In the tissues, they last about 4 or 5 days. Okay. That's because they're not going through the stress of going through capillaries and things like that. So circulation wears things down pretty quickly. Shorter timelines with infection, because if you have granulocytes taking part in infection, of course they're doing their job, and as soon as they do their job, they die. Monocytes and macrophages, circulating about a day, half a day to a day. When they go into tissues and stay and wait for things, they can last months or longer. And lymphocytes, they're constantly in lymph nodes, and they can last months or longer, okay, because they constantly want to be surveying things. Platelets, not the platelets themselves, but the megakaryocytes, they're replaced about every 10 days. All right, so that's a timeline about how long each one of these lasts. So if you had to compare a red blood cell to white blood cells, which one lasts longer? The answer is it depends, right? Red blood cells generally, but it depends, because it depends on where the white blood cells are located. All right. This is the fun part. How many of you guys feel like you understand ABO blood groups and blood typing? Some, a few. All right. So, here's what this is. We have a red blood cell. Ooh, I like this. We can use colors. All right. We have sugars on the surface of our red blood cells. And those sugars are what make up our blood type. Okay? Those sugars are what make up our blood type. We have, and those sugars are called antigens. Okay? We have the A antigen, we have the B antigen, and we have the O antigen. All right? The difference between them is, let's say that's the O. It's a short sugar. A antigen has one extended chain of sugars. A B antigen will have another extended chain of sugars. Okay? So, when we talk about how these work, the reason why A and B form a immune reaction, but O doesn't, is because of these longer sugars. Okay? That'll make sense in a few minutes. All right, another name for antigens is, bless you, agglutinogens. All right, because they cause agglutination. Do not confuse agglutination with coagulation. They are two completely different things. Students mix them up all the time. Don't do that. So, if these A and B antigens on the surface of red blood cells are sugars, how are they controlled genetically? Do our genes create sugars? What's a product of a gene? protein, not sugars. So how is our ABO blood genetically determined? No? What'd you say? Yeah. No? Good guess, though. Don't be afraid to say something. The enzymes that put the sugars on. Okay? So it's the enzymes that put the sugars on that determine our blood type. All right. So we have A, B, both of which are codominant to O. That's why you can have this result in an A blood type, this result in an A blood type, this result in an A blood type, but this results in an AB blood type. All right, if we're talking about from one copy of the gene to the other copy of the gene. Okay? Does that make sense? Okay. You know, the Punnett squares and all that stuff. We're not going to do that. Okay? You may. You may.
What about the RF?
We'll get there.
Okay.
We'll get there. So right now we want to refer to the ABO. All right? We'll do one really quick. Let's say mom has type A, but she's heterozygous with type O. Also. Actually, that's dad. Mom, I always say mom is always on top because that's how she likes it. Let's say mom has a B blood type, but she's BO. Okay? What kind of children could they make? An AB child, an AO child, which would be type A BO child, which would be type B, or an OO child, which would be type O. So with two heterozygous parents that are A and B, with an O allele that's recessive, you could potentially get all blood types for your children. All right? So that's the best example we can do. Does that make sense? So that's how the genetics of that works. Okay? That takes a 3-hour lab for undergraduate students. All right. Another term you need to know is agglutinins. Agglutinins are antibodies. And those are going to be the anti-A and anti-B that will attack A antigens or B antigens. All right? So the antibodies are mostly IgM and IgG. We'll talk about what those are later on. But if you are type B blood, you will have anti-A. If you're a type A blood, you will have anti-B. Okay? If you are type AB blood, you will have no antibodies. And if you are type O, you will have anti-A and B. Okay? So, the question is, if I'm type B blood, and I've never ever been exposed to type A blood, how do I have anti-A antibodies? Because we all have them shortly after we're born. Certain foods, bacteria, and environmental exposures will cause us to create antibodies against the opposite blood type. Okay? It's kind of like a cross-reactivity. Because bacteria, what's on their cell wall? Sugars. Okay? What is our blood type made from? Sugars. So there can be some cross-reactivity. So that's how we form these antibodies. All right. So this is a good summary of everything. This is a type A blood. This is type B. This is type AB, and this is type O. All right? So I like to draw, if you're type A blood, you have A antigens on the surface of your red blood cell, and so on and so on. I like to use the letters. All right? Again, if you're type A, you're going to have anti-B antibodies, and we just discussed why. If you're type B blood, you're going to have anti-A antibodies. We just discussed why. If you're type AB, you have neither. And if you're type O, you'll have both. All right? So, because type AB has no antibodies, they are known as the universal recipient. Universal recipient. So they can receive anybody's blood because their blood will not attack those red blood cells. Okay? Type O then, because they have both, they're considered the universal donor. So don't those antibodies that we're giving to the recipient attack their red blood cells? No, because we can get rid of them before we give it to them. Okay? 1, and 2, there's a dilution effect. The amount of blood you're giving to somebody is far less than the blood they already have. So those antibodies get diluted by a lot, and they don't have as much of an effect.
You said they remove the antibodies?
Mm-hmm.
Why can't they do that for A or B?
Your body has.
Your body has the antibodies attacking the blood cells.
Oh.
This is the blood that you're getting from somebody is going into your body, and so you can remove that blood, those antibodies. Does that make sense? Okay. All right. So, if we have somebody with type A blood and we give them type B, bad, right? Okay. If we give that, what's going to happen is the antibodies from this guy are going to bind to the red blood cells and cause clumping. Okay? That clumping of red blood cells from the antibodies binding to the red blood cells is called agglutination. Okay? That has nothing to do with thrombin and fibrin and platelets. Therefore, it's not coagulation. So this is just clumping. What's the problem with your blood clumping?
It's too thick to pump.
Too thick to pump, and it gets stuck. Okay?
So what was that process called? The causing clumping?
The what?
The causing clumping?
Agglutination. Okay, so this is kind of what that looks like. Here's what's happening. Here's what your blood looks like under a blood smear. Here's what happens when it's agglutinated. So all that clumping together. It's a mess, and it can be potentially life-threatening. Okay, so you have to make sure that you match blood types, right? Whoever you're getting, you have to make sure that it's going to match your blood type. So you look at the donor's antigen and the recipient's antibodies. All right? If the donor is type A and you're type B, which means you have anti-A antibodies, whoop, nope, you don't want to give it. Okay. Before we get to the Rh factor, if I have type A blood, who can I give blood to? Type A and AB. If I have type B blood, who can I give blood to? If I have type AB blood, who can I give blood to? Just AB. And if I have type O blood? All. Okay? I will ask questions on if you have if this is the donor's blood type and this is the recipient's blood type, will the transfusion react? Or if I have a type A person, what type of blood can I give that person? Can I give to? Okay? There will be a type of question like that. All right, the Rh antigen, rhesus, identified in rhesus monkeys originally. That's where the Rh comes from. This one, in order to have antibodies, you have to be exposed to the incompatible blood. Unlike ABO, this one you have to be exposed to blood previously. How do you get exposed to somebody else's blood? Birth? Not pregnancy, birth. It's the birthing process.
Is that why you don't have to give them the RH shot until their second kid?
Right. What else?
Transfusion.
Transfusions? Pardon?
Blood pact.
Blood packs. That weird but true. Yes. Sharing needles. Pardon? Eating a rhesus monkey. No, but that's fun. But that's funny. Okay. Now there are actually it's getting hot in here. Six Rh antigens, C, D, and E. They operate genetics very similar to like A versus O, dominant versus recessive. So there's a dominant C and a recessive C, dominant E, and so on. Okay, so these are the different potential genotypes. All right, the one we're concerned about is D. The D antigen is the one that is typically referred to as the rhesus antigen. C and E can produce reactions, but they're generally more mild, so we don't really worry about them as much. Okay, if the person has the D antigen, they are considered Rh positive. So genetically, if your genotype is big D, or big D, little d, you have the D antigen and you're Rh positive. If you have two small d's, then you're Rh negative. Okay? So, I'm going to go back real quick to ABOs. Why doesn't type A blood have anti-A antibodies? Oh! You're fired. Because you'd agglutinate your own blood. Okay? You and me. All right. So, if I'm Rh positive, am I ever, under any circumstance, going to have anti-D antibodies? No. There's no way, because then I'll be attacking my own blood. If I am Rh negative, will I ever have anti-D antibodies? It depends. Right? What does it depend on? Exposure. Good. Okay. When a person is exposed, they're said to be sensitized. So if you have an Rh negative mom who has an Rh positive child and you give birth, mom will form anti-D antibodies. So if you have a second Rh positive child, you have to get a shot of what's called RhoGAM. It's antibodies against the antibodies so that you don't attack your baby's blood. Okay? And that's what this is. Okay. If I have a naive Rh negative recipient, that means they've never been exposed. That means there will be no reaction. Actually, there will be a reaction. It's called a primary immune reaction. We'll talk about those. They're usually really slow and really weak, so you're not going to really have much of reaction. So it'll be delayed. Okay? But any future transfusions with Rh positive blood, then it's going to be a severe reaction right away. And as soon as that blood starts going into the patient, you're going to notice that patient start having some inflammatory problems going on. Okay, so that sensitization is really key. Okay, we talked about that. Any questions about that? So we have A, B, O, and RH.
So for RH, if the mom is positive and the baby's negative, would that count as a first exposure?
For the baby?
Yeah, for the baby.
For the baby, potentially. Yeah.
Does it pose a risk for the baby?
If that cord blood actually goes into circulation. Actually, no. No, it won't. It won't. Because the baby's blood never actually comes in contact with the mom's blood. But that cord blood from the baby will then mix with the mom's blood in the mom. Okay? So it could happen, but if it does, it's very rare. Okay, so if I am AB positive, who can I get blood from?
Everybody? Ever?
Yeah.
Because I don't have antibodies against anything, right? But if I'm AB negative, right, only negative ABO blood types. Okay, good. So what I would recommend is do some practice problems, do some scenarios. If you look in Canvas, I also have some breakdowns of what percentage of the population has what blood type, how these blood typings work. So there are some reference materials in Canvas for you guys as well. All right. If we don't go through all of hemostasis, I'm not worried about it because you guys are going to get this again in ClinMed. Okay? So hemostasis, stopping blood. Hemostasis, stop blood. All right? There's 4 mechanisms to do this. We'll go through them as we go along. What slide number is this? 55. Okay. So key events. We have a damage to a blood vessel. We stop bleeding. We form a clot. And then the clot goes away. Okay? So damage, clot, remove clot, go away. How does this occur? The first step would be a blood vessel spasm. So as soon as a blood vessel is cut, that smooth muscle undergoes an intrinsic reaction that clamps it off. And that automatically slows down blood flow, okay, to prevent blood loss. All right? Platelets that are getting stuck, that are in that general area, that are part of this damage, they'll release serotonin, and that will also stimulate that smooth muscle to contract. Okay? So it's an intrinsic response to the smooth muscle and platelets releasing serotonin. I thought I got rid of this. We already talked about what platelets are, so I'm just skipping over that slide. Yeah. All right. What do platelets do? Platelets have actin and myosin in there. What other tissue in our body has actin and myosin? Muscle. What do muscles do? So what do platelets do? Contract. Good. Okay, so platelets are capable of contracting. We'll talk about why that's important in a little bit. Platelets also have a residual ER and residual Golgi, so they are capable of making proteins and other factors that take part in the clotting process, including storing calcium, because calcium is absolutely needed for the coagulation cascade. And they contain mitochondrial enzymes to produce ATP for these enzymes to function. All right, platelet plug formation. What happens? We have an injured endothelial cell. All right, injured endothelial cells. What are endothelial cells? There's a lining on the inside of a blood vessel. Okay, so those injured endothelial cells will release something called von Willebrand factor. Von Willebrand factor is bivalent, so it will bind to collagen in the extracellular matrix, and it will also bind to platelets. So this is the first step in platelets adhering to the broken end of the blood vessel, von Willebrand factor. It'll bind to platelets and collagen. All right, once the platelets are bound, they're going to get bigger. They're going to get irregular in shape. They're going to look like jacks, ball and jacks. And that is going to cause them to interact with each other. So if they're sticky and they have fingers spreading out, they're going to stick to each other and form bridges. All right. They're also going to release platelet activating factor, which will tell other platelets, hey, you want to join us? There's a party coming on over here. And they're going to stick as well. And in the end, what you have is a platelet plug stopping blood flow. Okay? So blood vessel spasm, you slow down blood flow. Because you slow down blood flow, you're able to form a plug much easier. All right? Then we have clot formation. Or as Cody would say, agglutination formation. Just giving you crap. All right. So clot formation begins within 15 to 20 seconds. Okay, 15 to 20 seconds. So it is a really fast process. And you can have a completely blocked blood vessel with this clot within 6 minutes. Okay? Unless your abdominal aorta bursts. Okay, then you're just going to die. If you're in the middle of the country and no hospitals are around. That's rural health, folks. All right, within an hour, that clot will retract. Okay, it will retract. And then within 1 to 2 weeks, you're going to have fibroblasts lay down more collagen and form scar tissue. Hopefully, endothelial cells will also migrate to the area and make it a functional repair as well, but that depends on how big much damage there is. Okay, so with blood clotting, this is the big thing to know. What happens is you have prothrombin activator, converts prothrombin to thrombin. So this is very much like those zymogens, those digestive enzymes. You convert one inactive factor to an active factor. And notice that calcium is involved for almost every step. Okay, so prothrombin activator is formed, convert prothrombin to thrombin. Thrombin converts fibrinogen to fibrin, and a fibrin forms this net-like structure that reinforces that platelet plug. Okay, so that fibrous network that reinforces the platelet plug. Okay, it turns out, go ahead.
How is the calcium involved? Is it just being exchanged, or?
No, calcium is needed for those factors to work properly. Yep. So calcium, a cofactor. Call it a cofactor. Okay? The cool thing about clots, one of the cool things, is that they are a positive feedback mechanism. One of the few positive feedback mechanisms you'll see. Once you form thrombin that's trapped in the clot, that will convert more prothrombin to thrombin. So you don't need prothrombin activator anymore. Does that make sense? So once you form thrombin, then you convert a lot more prothrombin to thrombin, positive feedback mechanism, and the clot gets bigger and bigger and bigger. Okay? And then not only will that make the clot bigger, but it will extend it past the site of the injury, anchoring and reinforcing where the clot is located. All right? We said that the clot contains contractile proteins. Okay? I'm sorry. What does platelets contain? Contractile proteins. So within about 20 to 60 minutes, the platelets that are within the clot, they're going to start ratcheting on each other and bringing things closer together. Okay? Bringing blood vessels, damaged blood vessels, closer together. So that's what we call clot retraction. Right here. All right? That clot retraction is going to tighten the area. Serum is going to be pushed through. Why serum? Because you got rid of all the clotting factors in the area, so now serum is going to be circulating. and the vascular defect is closed. Now that you've closed the bleeding problem, now you can repair with the fibroblasts, with some new smooth muscle cells, with new endothelial cells. Okay? And this is what a retracted clot looks like in a test tube. Okay. I know everybody loves this, right? Intrinsic and intrinsic clotting pathways. You have two pathways. We'll keep this simple. Intrinsic, everything that's needed to form the clot is already in the blood. Extrinsic, you have to have a factor from the outside activate the clotting pathway. Okay? So this is a summary slide of everything, so I'm not going to go through the whole thing. Calcium is required for most of the processes as a cofactor. But looking at this, which is the more complicated pathway? The intrinsic. So which do you think is going to take longer? Intrinsic. Okay? And this down here is that pathway we've already talked about. That's called the common pathway. All right? So both pathways converge onto this, and both of them activate the same common pathway. That's why it's called the common pathway. This is just a list of all the clotting factors, what their number is, what their name is, what type of molecule they are, where they come from, and what pathway they're involved in. If you're like, I can't keep these clotting factors straight with each other, this is a good table to go back to. All right, intrinsic clotting mechanism. We'll just start with the picture. Okay, you have some sort of blood trauma, let's say even turbulent flow. Remember, turbulent flow, that non-laminar flow, can be help form a clot. So that's what that would be. Or let's say that Gavin was talking yesterday. It's like, this pipe, where does the gunk form? On the surface. In your blood vessels, it forms under, right? He was talking about that. So that would be another example of inside the blood vessel. All right? That's going to activate factor 12. Factor 12 is going to activate factor 11. Factor 11 is going to activate factor 9. So what do you notice happening?
It's going down.
Right? It's going down.
Give 10, but that's okay.
Okay? That's going to activate factor 10. And then 10 is going to be part of the prothrombin activator that activates thrombin for the common pathway. Okay? So it's going to go down 12, 11, 9, 10. Extrinsic, on the other hand, you have some sort of tissue trauma, you release a tissue factor. You're going to see in a previous slide that's called Hagman factor. If you look back a few slides, you'll say Hagman factor. That's the tissue factor. That's going to activate factor 7, which is then going to activate factor 10. So from there, the pathway is the same. So if you go back to this overarching picture, they really converge at factor 10. Okay? Extrinsic goes from 7 to 10. Intrinsic goes from 12, 11, 9 to 10. Technically 9, 8 to 10. So that's an easy way to remember the clotting factors that are involved. Okay? When you get to this part, and I want to focus on this because this is the most important, this common pathway, That's the rate limiting step of the pathway, the prothrombin activator. As soon as that's formed, the faster that's formed, the faster your clot can form. Okay? In the end, what you have are fibrin fibers, and these, each one of these here is a fibrin fiber. Notice they all get linked to one another to form a fibrin mesh. So there's literally a net over these platelets. Okay, that's the end product. This is just, we were in Florida a couple years ago in Tarpon Springs. And that's known for their sponges. They're known for their sponges. They call themselves the sponge capital of the world. It's a Greek community. Really cool. But they had these boats, and they had sponges in nets. And what does your physiology professor first think of? That looks like coagulation to me. So I took a picture and put it in the lecture. So literally, imagine a sponge as platelets, and the net surrounding them is your fibrin mesh. And that's stopping blood flow. Okay. What do I want to say about this? Even though we have two separate pathways, can both be initiated at the same time? Of course. If you have injury to a blood vessel from the outside, you're going to release tissue factor, but that intrinsic pathway is also going to be activated as well. Okay? So extrinsic pathway can be very explosive, clotting within 15 seconds. Why? One factor is involved, and then the common pathway. Whereas with intrinsic, it's like, we'll just take our time getting there. So that's why the intrinsic pathway is much slower. And if you think about that, that is orders of magnitude slower. Not just slower, much, much slower. Okay, vitamin K, important for clotting. Why? It's actually produced by bacteria. We don't produce this by ourselves. Vitamin K, fat or water-soluble vitamin? Fat. Good. Actually, it's right there. Jerks. So, it's important for producing clotting factors. Okay? One of the things it's important for is producing clotting factors, and it's realized to be so important for this process that that's why we give vitamin K injections to infants. Or if patients have liver or biliary disease, they give vitamin K before surgery. Okay, so just know that it's important for the formation of clotting factors. I'm not going to go into the details of that. I won't ask you about any of that. All right, but we have this clot formed. Do we want that clot to stay there? No, because then we'll prevent blood flow. So what happens is another circulating protein called plasminogen gets trapped in the clot. Circulating plasma protein that gets trapped there. Plasminogen suggests that it is an inactive form. After several days, injured tissues, those endothelial cells, they'll release something called TPA, tissue plasminogen activator, that will activate plasminogen, convert it to plasmin. Plasmin will start to digest that fibrin. And once you start cutting the nets, what's going to happen to all the sponges? They're going to be released, and they're going to go back into circulation, right? Most likely going to be destroyed at some point. Okay, so that's going to result in reopening blood vessels. Okay. You guys can watch this video. Basically, it just shows this milky fluid. You put TPA in there, and that milky fluid disappears to a clear fluid. So you take a insoluble protein and you solubilize it because you convert it back into fibrin monomers. Okay? Clot lyse fibrinolysis. So this process is called fibrinolysis. Restores blood flow. It also produces something called D-dimers. That is a really important diagnostic tool because if you see an increased D dimer level in your patients, that probably means that they had clots forming abnormally somewhere, DVTs maybe, and you need to address that issue. Oh, sweet, look. It's actually playing. Watch. It's exciting. All right, it's just not as exciting as I make it out to be. It's like watching grass grow. For all I know, it could have stopped. There, you can see it clearing up. So that's a fibrin clot digested. I thought it was cool. Kind of brings the point home. All right. When a clot forms abnormally, it's called a thrombus. When that thrombus breaks off and starts circulating and blocks blood vessels, that's called an embolus. Okay, so if you look at your IOs, what's define clot, define thrombus, define embolus. Okay, these are your definitions. All right, so we have all these clotting factors in our blood. Why aren't we clotting all the time? All right, there are a lot of things that prevent it. One is the moving around. Another is the smoothness of the endothelial surface. You glide across a smooth surface, you're not going to have turbulent flow. All the cells also have the glycocalyx, which makes them really smooth also. We have proteins that compete for thrombin. So if thrombin actually gets formed, these thrombomodulin molecules will bind them up and prevent them from abnormally forming a clot.